Hello and welcome back to Honors Biology. So today we're going to be really starting off and talking about our next unit, the cell cycle and mitosis. So really this is going to explain how do we go from one cell, right, our zygote, how do we go from our zygote to, a human being that has trillions and trillions of cells. What does that process look like? Well, it's cell division. Cells just divide over and over and over again. So that's what we're going to be exploring in this unit. So before we get really going too far, just some things just to keep in mind as we talk about these cells. So we talk about somatic cells. We talk about gene editing. We talk about somatic and germline cells. This matters here. So somatic cells, they're body cells, right? So they're produced through a process called mitosis, and then they have 46 chromosomes or 23 pairs. So that 23 pairs of chromosomes for a total of 46 chromosomes. Then there's a thing called homologous chromosomes, which is each member of a chromosome pair. So in each pair, we have homologous chromosomes. And then down here, diploid and haploid just refer to the number of chromosomes. So we have diploid cells. So all of your somatic cells are diploid cells. They're called 2N, and because they have a total of 46 chromosomes. In people, in each different animal, in each different species, the number of chromosomes change. Flies have a different number of chromosomes than in frogs or in deer. There's a number of different chromosomes there. So that's all our somatic cells have that. And then we have haploid cells, which have half the number of chromosomes. And those are just represented just with an N. So in people, instead of having 46, haploid cells have 23. And those are found in the gametes or the sperm and egg cells. So our sperm and egg cells have half the number, so they're haploid. So let's just talk more about chromosomes. So that's what we're talking really about, is how do the chromosomes divide here. So the chromosomes is how we pass on information. Okay. Before cell division, each chromosome has to get duplicated. And how do we get a chromosome? Well, we get a chromosome by just compacting the DNA. Remember, there's 6 feet of DNA in each cell. So when a cell goes to divide, it can't have all that DNA just floating there. It has to compact it. So really it just folds and wraps and super folds again and again and again until we get this classic chromosome structure. So this is what a chromosome looks like unduplicated. Before it's replicated, this is what it looks like. And then when it duplicates, we get that classic X kind of shape that we look like here. So this is a different version, right? This is another picture showing you that there's one chromosome's unduplicated, and then when we duplicate, we get these chromosomes here. All right. And each strand here, each kind of one chromosome, we call this a chromatid. So this is one chromatid, and this is one chromatid. And since they're identical to each other, we call them sister chromatids. So the chromosome is made up of sister chromatids. So each chromosome has 2 identical sister chromatids, right? When DNA replicates, okay, it creates an identical copy during replication. So each chromatid is the same, so we call them sister chromatids. And then they're joined in the middle. Well, it's not always in the middle, but they're joined together at this point, in the point where the sister chromatids join together, kind of like where the X meets, we call that a centromere. So this middle part here, this is the centromere of a chromosome right here. So it's important that we kind of review chromosomes and what they are, because mitosis and meiosis, the cell cycle does a lot with chromosomes and the structure of them. So let's talk about the cell cycle now. So the cell cycle, all it is a sequence of growth and division for a cell. There's two different phases for the cell, interphase and the M phase. In somatic cells, that's mitosis. In gametes, so your sex cells, that's meiosis. But we're just focused on just the cell cycle, just that interphase part right now. So 95% of a cell's life is going to be spent in interphase, and then 5% of a cell cycle is going to be spent in mitosis. So most of a cell's life is spent in interphase. So what does that look like? Well, this is what it looks like. All right. Interphase is made up of three different parts, G1, S, and G2. And we'll talk about what happens in each of these phases, but collectively, these three phases are known as interphase. So interphase is made up of G1, S, and G2. And then the M phase for somatic cells, for body cells, it goes through mitosis, and mitosis has 5 steps to it: prophase, metaphase, anaphase, telophase, and then cytokinesis. I have a whole separate video on mitosis, so we're just worrying just about this first interphase right here. So let's take a closer look at these 3 different phases that make up interphase. So what those letters mean, G1, S, and G2, is they're just acronyms. So we got growth stage 1, so G1, the cell's growing. Synthesis stage, which is the S phase, that means the DNA is replicating. And then growth stage 2, we're just preparing to divide at that point. So during G1, what's happening during growth stage 1, there's really 2 things. The cell's increasing in size, and we're making new proteins and organelles. Because when we divide the cell, we also have to, make sure each cell has enough mitochondria or chloroplasts in plant cells, or Golgi apparatus, the endoplasmic reticulum. We have to make sure that there's enough of that to go for both cells. So we have to make all these new things to feed both cells. So that's happening during G1. S phase synthesis is just that the chromosomes replicate. So this is where DNA replicates takes place. And you know how DNA replicates, right? Helicase comes in, breaks apart the double helix, polymerase comes through, it's going to replicate the leading and lagging strand. In the end, we get 2 new DNA molecules. So we get 2 new chromatids that join together to make a chromosome. Once a cell, so cells can hang out in G1 for a very long time. For some cells, each cell is divided at a different rate, but cells hang out in G1 for most of that time. Once a cell is going to enter S phase, once it goes through S phase, it passes the point of no return. Once we replicate the DNA, and once we have all these sister chromatids lined up, we have to complete the rest of the cycle in. So once we enter, then most cells are going to finish the cycle out. And then right before we enter this M phase, this mitosis part, we go through G2, which is second growth. So we replicate, all the organelles and molecules required for cell division are produced. So we'll talk about in mitosis of centrioles and spindle fibers and how those get made. Those are made during G2. And then once G2 is complete, once we have everything on deck, well, then we go through mitosis. Mitosis is just the division of the nuclei. So that's when we split up the chromosomes and we split up the nucleus. So I have a whole separate video for mitosis. So that's it for the cell cycle and chromosomes. Let me know if you have any questions, and I'll see you next time in class. Bye.